want to turn viruses on their end and make them truly beneficial. You may remember we started off way in the beginning talking about how probably most of the viruses infecting us are good. We're not sure how, but we can actually turn them for the good, and that's what I want to talk about today. I want to talk about viral gene therapy. What do I mean by this? I mean using a virus, which we give it the term virus vector because we're putting things into them. We're going to use virus vectors for a number of purposes.
You can see how the hind wings are stored, folded up like an accordion.
To do actual gene therapy, which means we give patients a gene when they're either missing it or they have defective versions of it. So we can put the normal gene into a virus vector and deliver it to the patients. And that is what most people think of as gene therapy. But I also put these other categories under gene therapy as well to deliver an antigen, as in a viral vaccine. We've mentioned some of these already in this course, but they fall under viral gene therapy because we're using vectors to deliver a part of a virus. We can use viruses for doing what we call viral oncotherapy, that is to kill tumors. And we can take two approaches here, as you will see. We can use unmodified viruses, or more likely and more frequently now, we can insert additional genes into the viral genome to make them more effective at killing tumors.
Sometimes in big groups.
And of course, vectors have huge uses in research outside of the practical applications that we're going to talk about today. So what I would like to do is go through a number of examples of each of these categories and show you how these are actually being used today. It all starts here. This was something we talked about, I think, in lecture 2 when we talked about methods, and that is making infectious DNA copies of viral genomes. And this was first done in the 70s, first with bacteriophages, then with animal viruses, to convert the genome into a DNA copy for RNA viruses, or to take the DNA of the virus that's already a DNA virus and put them in plasmid vectors so that they can be modified, because the techniques of recombinant DNA, which arose in the 70s and 80s, can be used to modify genomes in terms of putting in therapeutic genes or removing genes that you don't want to be there. So this is the example I show with poliovirus, where we can take the viral RNA and make a DNA copy of it using reverse transcriptase. And this is the power of having that enzyme, reverse transcriptase, which can be used to make DNA copies of RNA genomes. So now we have a copy of poliovirus DNA in a plasmid, which can be grown in bacteria. And then we can take that DNA and introduce it into cells, and that will initiate an infectious cycle. That's enormously useful, of course, because it allows you to modify the viral genome. So for every viral vector that we talk about today, this technology, the infectious DNA clone, is used to get the final end product. Let's talk about a few of the different kinds of viruses that are used in viral gene therapy. We're going to touch on some of the key players that we've talked about in this course so far.